Hello everyone, my name is Darren Seidel, and it's a pleasure to be with you today. I'm presenting on a particular topic for this semester's microbial ecology of the ruminant class that's really important to the future of my dissertation, and that would be monoterpene effects of pure culture pathogen and mixed culture ruminant fluids, to seeing the dynamic changes in microbial performance when inoculated with monoterpenes and what those particular anti-nutritional properties do to some of the microbes inside the ruminant that are found throughout the GI tract. So today I would like to really touch base on the context of what exactly is a monoterpene. I feel like it's essential for me as a presenter to catch the audience up on some of the basis for what it is that I study and why is it important, those kind of rhyme or reason things. So terpenes are derived based of isoprenes, and they have different structures based on their isoprene combinations. So monoterpenes in particular are two isoprene units smashed together with a 10 carbon base, and then there's a little bit of variability past that point. So as you can see, most of them, if they would truly fit the isoprene unit dynamic, you would have 10 carbons and then 16 hydrogens, but there's a little bit of variability in those, particularly with what we can see in camphor to the far right. It has an extra oxygen tied into that bottom right of that particular structure, and then halogen, which is a unique monoterpene, a very, very odd essential oil in a way that's produced by algae, and you can see the additional anions on top of that, and then only 15 hydrogens, but the carbon base unit should always remain the same. And then essential oils, there's this headbutting that you can look at through the literature as to what is the true mechanism by which animals are deterred away from plants, and one of those arguments is essential oils, and another big one is condensed tannins. But for the essential oil principles, their defense mechanism, but they also alter microbial activity, and documented in several publications. The fact of the matter is, what is truly happening is that the plant that's not palatable to the animal is why it's deterred away, or consuming the plant doesn't make it sick. And the really kind of mind-busting thing within this particular topic is not just one set of plants have a exact percentage of essential oils, and that makes it tough from the research side of things, because percentage variability can change based on maturity, seasonality, plant parts, that meaning the leaf versus the stem in cases, region, human alterations, whether we're chemically trying to kill the plant. If you kill the plant, of course their essential oil profile would be 0. Hopefully I get a little chuckle out of that. But moving on, it's one of those unique things where the literature is stating that you can do a particular study for a particular plant at a particular time, and you can try that on the same plant, and the repeatability of that varies a little bit from the essential oil profile side of things. But one particular plant species of interest of mine is within the juniper species, and particularly two within those, the pentacoti and the achaea, which their common names, as you can see in the next slide, are redberry juniper and blueberry juniper. So just like naming bacteria, whoever named these plant species didn't get really creative from a common name side of things, because the distinguishment between the two is based on their color of their berry. And why is this important? Okay, so juniper encroachment in West Texas has been an ongoing battle for sheep, goat, and cattle producers in the Concho Valley and further in the Trans-Pecos region for a number of years. What juniper does is it encroaches, it chokes out rangeland, and potentially has the ability to alter how many animals you can run within a particular area. So in the bottom picture is within Crockett County, Texas, in an overlook, and it might be hard to see, but trust me, the only thing green in that particular picture is actually juniper. So you can imagine what a rancher has to work with in some cases, and the unique evolutionary design of the plant is a whole nother topic I won't touch on, but its maturity. So in a mature state, it has a different essential oil profile than an immature state. And just to go further, that's the difference between 3 meters one way or the other. And then the leaves from a fresh side of things, you can see the variability within publications on truly what's going on in their essential oil percentage as well. So kind of a running concept, a running theme of there's not just one answer, and it's hard to control this for a particular experiment when your plant has that much variability as well. So the two punchlines of this, we talked about monoterpenes and we talked about the plant. Well, from my side of things, I truly am interested in what we can do as an industry to progress our antimicrobial, antibacteria usage, and to kind of develop a new mindset of stewardship. So every producer from an animal production side of things is trying to do what the consumer wants them to do, and consumer perception right now is trying to tell us to steer away from antibiotics, even though that we try to do the best we can with ensuring that the animals remain healthy for a wholesome food product. But what if these essential oils can lock a natural extraction process that we can use to alter microbial performance to even better our production standards? So I put this up there because this is the current press right now, and this is what everybody's trying to steer towards or get away from in the grocery store, even though most of the people making those decisions or those perceptions might not have an agriculture background whatsoever. So I think essentially for a microbial ecology of the rumen class, we can profile what kind of bugs are truly going on inside of the rumen, and then we can also look at the pathogens that I want to work with too. So if you have not done it, Dr. Calloway in this class has mentioned this book, but please don't let the time go by and not read it. Dr. Russell does a great job of making it easy to understand the ruminant microecology, the ruminant microbiology as a whole, in a really compact version of his book. So please look at that. As you can see, some of the starch and sugar utilizers, the fermenters from the forage side of things, and then my pathogens of interest. Newport has made the current news as being the most recent outbreak influenced through the JBS ground beef scare that happened last month, so not too long ago, and you can see the CDC link to more information of that. But from a pathogenic side of things, it is present in the ruminant, it's present in ruminant animals. Salmonella is two types of interest for my particular research, for Newport and Typhimurium, and then the big one everybody wraps their head around, E. coli 015787, and those are just two of the biggest outbreaks throughout its long history. So, report objectives, and I'll kind of touch on this and really hammer home the fact that camphor has been one of those particular monoterpenes in ongoing research that we've looked at, but now that we have preliminary data and information that's showing a little bit of alteration, and then preliminary research that shows particular threshold from a pure culture side of things, what can we do to potentially ensure that maybe there's other monoterpenes that have the ability to do the same thing that camphor has done from a limiting or a threshold side of things. So three of those particular interests of mine from a monoterpene side of things are sabinine, limonene, and boryl acetate, and the relative nature of those is their high percentage within the percentage of total essential oils within these juniper species that I previously mentioned. So we'll see where this goes. It's really unique. We can get a baseline for sublethal and lethal type thresholds, and then maybe be able to spin that off on truly what's happening in mixed cultures when we don't know all the organisms within a particular media. But it's a unique thing to look at, a unique thing in my opinion, and I hope you found this presentation interesting as well. Thank you for playing along, and I look forward to hearing all of your pros, cons, and anything that could help me further on. Thank you for your time, and you have a great rest of your day.